So our last video left off right before we jumped into mitosis. So again, we've got 4 steps here. We're going to look at what's happening to the DNA, the nucleus, and then the spindle fibers as we're doing this. So as with both mitosis and meiosis, we're going to be preceded by interphase. Okay. And again, interphase is where that cell is growing. It's getting ready for cell division, and we're replicating our DNA in that S phase of interphase. Okay. And so we've got that gap 1 where it's growing, the S phase, DNA synthesis, we're replicating our DNA, then gap 2 where it's preparing for this division. Okay. Then we can jump into mitosis. So here we see our centrioles. Okay, and we're jumping into mitosis now and into prophase. So our very first part of mitosis, the first phase, is prophase. Things that we see happen. Normally in the cell, the DNA is in the form of chromatin. When we get into prophase, we start having that DNA come together, and it's going to go from chromatin into the form of chromosomes. Okay, so they condense so that they take up less space, they wrap around proteins. But the big thing here, the DNA goes from chromatin to chromosomes. We start building our spindle fibers from our centrioles. Okay, and then the last thing here is we start breaking down that nucleus. We can't do a whole lot with the DNA until that nucleus is gone. So those are the 3 things that are happening in prophase. After prophase, we jump into metaphase. Okay, this prefix in this case, meta means middle. So all of our chromosomes are going to line up down the middle of the cell. And they're going to do this because of the spindle fibers that have attached to the centromeres of those chromosomes. Okay, so metaphase, all the chromosomes are going to line up in the middle. The third phase is anaphase. In anaphase, we're going to separate those chromosomes. So the spindle fibers are going to start to shorten. We're going to separate the chromosomes at the centromere, and they're going to start moving to opposite sides of the cell. Okay, so anaphase, we're separating our chromosomes. They're moving to opposite sides. And then our 4th and final phase of mitosis is telophase. Okay, telophase is the opposite of prophase. So in telophase, our chromosomes are going to turn back into chromatin. The nuclear envelope is going to start to reform. You can see that starting to come back. And then those spindle fibers that we saw in our other phases have started to break down, and they're going to disappear. Okay, and so that's telophase. And then, of course, we've got to follow this up with the process of cytokinesis, where these cells go from one cell and separate into 2 separate cells. Okay, and that's mitosis. Okay, so these images are probably what you're going to want to look at when you do that stop motion for the process of mitosis. Okay, so when we look at cytokinesis, what is actually happening? Okay, when we're looking at animal cells, they're going to produce a cleavage furrow. Basically, we've got microfilaments that are going to start to surround the center of that cell. Okay, and this is like a drawstring bag, and they start pulling on that string tighter and tighter, and so that area gets smaller and smaller. Okay, and so what we see is eventually it gets so small that they just pinch off, and they are completely separate cells now. Okay, but we see that we have this cleavage, and that's what this is named for, is we get this cleavage furrow forms between these cells. They pinch off into 2. So that's cytokinesis in animal cells. Plant cells have a slightly different method. Because they have a cell wall, you really can't pinch off to make new plant cells. So instead, what we see happen is we start to form a cell plate. So we have our anaphase where we separate those chromosomes. Here we've got our telophase, the nucleus reformed. And during these phases, we start to make this cell plate. Okay. And this cell plate forms more and more, and it gets bigger and bigger until we get this complete cell wall separating these two cells. Okay. And this is what we see happen in plant cells. Okay. And so here we're just seeing this again. Cleavage furrow in animal cells for cytokinesis, new cell plates that are going to form a brand new cell wall in our plant cells. Okay, and so this shows this whole process of mitosis. And so here we've got prophase here, break down that nucleus. We line them up in the middle, metaphase, separate them, anaphase, telophase, make that nucleus, and cytokinesis, separate them. It happens really fast in this video. You can watch it a few times. You can see this entire process of mitosis happen. Okay. What do we get through mitosis? Two genetically identical daughter cells. So the two cells that we get out of this process are exactly the same. Okay. They are also diploid, so they have a full complement of chromosomes. We show this as 2N. And what this means basically is in human cells, we've got 23 pairs of chromosomes for a total of 46. When our cells go through mitosis, we're going to end up with two cells that each have a total of 46 chromosomes. They're the exact same as those parent cells. They're clones of one another. Okay? We see that some organisms will reproduce through mitosis. We already saw that bacteria do. Well, it's not quite mitosis, but it's similar. But we see aspen trees do this as well through their root systems, where what we think are different individuals growing are just reproductions through mitosis. And so here we see in these forests, this whole area of these trees are the exact same tree, and they're growing through mitosis. We're not getting any sexual reproduction here between these trees at all. Okay? So things that you want to make sure that you know, and the answers are already here in a face-to-face. We talk about this separately. But why do cells undergo mitosis? Again, to grow, repair. Reproduction doesn't quite fit here with mitosis. But if it is asexual reproduction like those aspens, then yeah, they do reproduce through this process. Okay. Events of each stage of mitosis. So you need to know the order: prophase, metaphase, anaphase, telophase, and then what's happening in each of these phases. Okay. And then at the end of this process of mitosis, what do we get? Two genetically identical diploid cells. Okay, so we'll end there for mitosis. We'll pick up in the next video here with meiosis.